So 2024 was a very active year in research on antimicrobial resistance. This year was also highlighted by a high-level political declaration by the United Nations, putting antimicrobial resistance as one of the most important healthcare problems in the world that needs important attention. To start 2025, we are having a very new podcast this time, in which I have the honor to have invited members of trainees of the antimicrobial resistant T32 NIH-funded training programs around the country. From the Texas Medical Center here in snowy Houston, we're actually recording with several inches of snow in Houston for the first time. University of Pittsburgh and Emory University, which are sort of programs that are pioneers in trying to train young talent and bring young talent to antimicrobial resistance. So welcome to Editors in Conversation, to the first podcast of 2025. The objectives of this podcast is to review, discuss, and highlight the best paper published in AAC in 2024 in gram-positive, gram-negative, mycobacterial, and parasite resistance. This episode is brought to you by Antimicrobial Agents and Chemotherapy Journal, available at asm.org/AAC. I'm your host, Cesar Arias, Editor-in-Chief of AAC. And this podcast is supported by the American Society for Microbiology that publishes AAC. If you plan to publish in AAC, ASM members get up to 50% off publishing fees. And I hope you become a member soon if you are not already one of the largest scientific societies in the world. Visit asm.org/membership to sign up. Don't forget to check our latest issue of AAC, available at asm.org/AAC, with outstanding papers on mechanisms of resistance, pharmacology of antimicrobial agents, epidemiology, and clinical therapeutics, amongst others. And particularly today, we're going to highlight certain papers that you can go there and check those outstanding papers. We have several sections now very well established, including challenging clinical cases, new perspective, antimicrobial agents, and a section on antimicrobial stewardship. So I also have the honor to have here the directors of these training programs at Emory University, Dr. William Schaefer, and Dr. Harrison Lee, who is the director of the program at the University of Pittsburgh. And joining us are these outstanding trainees. You're going to hear them, but it's going to be Nathalie Chen from the University of Pittsburgh, Alice Kang from Houston Methodist Research Institute, Nathan Raabe with University of Pittsburgh, Taylor Blackburn, Emory University, and Jacob McPherson from the University of Houston in Houston, Texas. So welcome everybody to the show. The first time we have so many guests in one show. So this is going to be fun. Right. So we're going to start by discussing these papers on this. So we're going to start with Nathalie. Nathalie is going to discuss the first paper on gram-positive resistance. So Nathalie, go ahead and tell us why you choose this paper, why you like it.
Sure. So the paper that I'll first start talking about is titled Bacteriophage Therapy Reduces Staphylococcus aureus in a Porcine and Human Ex Vivo Burn Wound Infection Model. And this was some work done by Michel Molendiet and others from the Erasmus University Medical Center in the Netherlands. And so in short, in this work, the authors aim to determine the efficacy of bacteriophage therapy for burn wounds infected with Staph aureus. And phage therapy has been a really hot growing field as a promising alternative therapy to antibiotics. Some previous clinical trials have been done testing phage for burn wound infections, but unfortunately those failed due to a couple different challenges, primarily being associated with phage manufacturing or logistics of phage administration. But there's been promising data in animal studies, and the authors hope that their work would help establish some more guidelines for how phage could be potentially used in burn wounds.
What did they find? Tell us what the main findings of the paper.
Yeah, sure. So they tested a couple different variables here. So they, like the title suggests, they used two different ex vivo models. So they used a human and a porcine one, and then they used two different types of phage to compare those two, and then they compared phage therapy with fusidic acid as kind of an antibiotic control. Fusidic acid is often used for staph-infected burn wounds. And then they tested three different concentrations, and then therapy with three different types of methods. So they tested a single application of phage and then looked at 2, 4, and 24 hours after treatment to see how much bacteria was left in these different wound models. And then they also tested a successive phage application model where after the burn wound was infected, they treated with phage three times, each 3 hours apart for a total of three treatments. And then the last type of treatment was a prophylactic model where before they applied bacteria to the wound, they would prophylactically treat with either the phage or the antibiotic fusidic acid, and then tested how much bacteria was left in that. They found that the prophylactic model worked the best. There were differences between the human skin versus the porcine skin, which they got into a little bit in terms of reasons why that might be. But overall, it was it was a little bit challenging. They didn't have a ton of efficacy at every single step. With the phage treatment, they did, however, find that phage was a lot more effective, at least better, like more effective than fusidic acid, or at least equal to fusidic acid in each of these trials. And so I thought that was really cool. And I'm really interested in phage therapy. And so this was like an ex vivo model, but I think it could really help down the line for when they start putting these into people doing clinical trials, just getting a sense of, what types of, like, what ways should we be applying phage, how much phage do we need, and stuff like that.
Oh yes, phages, and phages. Seem to be is something that people are using very commonly. It is very difficult, really, to find clinical data supporting good efficacy yet. But I think those studies are coming. So, Bill, Lee, any comments about this? There is a lot of interest, of course, in phages, but finding true efficacy is becoming very challenging. And this paper was very well done, where very strict methodology shows that it's feasible, but it's not a home run. What are your thoughts?
Yeah, so I actually really enjoyed this paper. And one of the things I think it does emphasize is possible host restriction matters in using different models, with differences that one might get with the porcine versus human ex vivo model. But yes, it was certainly very well controlled and does give us something to look forward to.
Yeah, and I agree. Excellent paper. Phage therapy has been one of those things that's been the wave of the future for a long time. So we're not quite there yet, and I agree there are limited data on efficacy, and there are trials being in the works. And I think that those are the data that we're really going to need to see to determine whether this is a therapy that's going to work, going forward.
Okay, great. Thank you, Nathalie, for highlighting phage therapy. Let's move on. Alex, he's going to talk about what paper on gram-negative resistance. Tell us, what paper did you choose and why?
Thank you, Cesar. I think the first paper I chose is titled Relative Inhibitory Activities of the Broad-Spectrum Beta-Lactamase Inhibitor, Zerborbactam, in comparison with taniborbactam against metallo-beta-lactamases produced in E. coli and Pseudomonas aeruginosa. So this paper was looking at the efficacy of these two up-and-coming new agents. These are chemicals that were developed to overcome the issue of metallo-beta-lactamases, which are enzymes that can break down a major class of antibiotics called beta-lactams. And so we really don't have a good clinical solution to these metallo-beta-lactamases. So these two novel agents are an exciting solution to that. So this paper was comparing the efficiencies of zerborbactam and taniborbactam in E. coli and Pseudomonas strains that have been recombinantly engineered to produce various metallo-beta-lactamases. So they had E. coli and Pseudomonas strains that produce various variants of NDM metallo-beta-lactamases, as well as the VIM metallo-beta-lactamases and the IMP metallo-beta-lactamases. And they used standard antimicrobial susceptibility testing, broth microdilution testing, to test the MICs of various cephalosporin antibiotics, ceftazidime and cefepime, along with the carbapenem, meropenem, in combination with these novel beta-lactamase inhibitors, again in Pseudomonas and E. coli. And some major findings is that if you compare these two novel agents, it seems like, at least in E. coli, Zarabrobactam far outperforms Taniborbactam for nearly all of the beta-lactamases that were tested and expressed in E. coli. And importantly, that included the IMP enzymes, which previously have been shown to evade Taniborbactam. So that was a big issue with that compound, but Zarabrobactam kind of fixes that issue. But it seems like even Zarabrobactam is not perfect. They have found a variant of IMP10 specifically, that was not effectively inhibited by Zarabrobactam. In Pseudomonas, we actually see an opposite effect where Taniborbactam does better than Zarabrobactam. And the authors believe that is because Pseudomonas has a lot of multi-drug efflux activity. So that is when an antibiotic is pumped out of the bacteria by these various efflux pumps. So it seems like these beta-lactamase inhibitors are also a substrate to drug efflux, which is, and the authors believe that, and they support that with testing various mutants in multi-drug efflux, that might be a downfall of Zarabrobactam in Pseudomonas.
Okay, beautiful presentation. Thank you very much. So 2024, we get closer and closer, and this is, I think, the consolidation of these broad ultra spectrum beta-lactamase inhibitors, which are very promising for us due to the enormous problems with resistance that we have. I think this is an example how complicated this field has become and how important it's for clinicians to understand the subtle difference between these inhibitors, that when they are going to become available. Hopefully some of them will become available in the next 2 years. So Lee, any comments on this?
Yeah, I mean, the agents sound very promising based on the data so far. I think what we find is that as soon as we develop new agents and start using them widely, we start to see resistance. It's almost inevitable. And so I think these sound promising, but it's going to be a constant development of new agents to keep ahead of these bugs.
Yeah.
Yeah, Bill?
Yeah, that's exactly right. I'm glad Lee said that. It also points out the importance of drug efflux to recognizing these inhibitors, and also points out that one size will not fit all. So we have to know about the bug, and we have to know about the inhibitor. We have to know about drug efflux. So it's very, very complicated, but we have to move forward.
Yeah, and clinicians need to understand this very importantly, because we are the one who are deploying these drugs. So AAC will have a lot of roles to try to teach us what to do with these drugs. Excellent, Alex. Let's move on to Nathan. Nathan is going to talk about fungal resistance. Tell us about your paper.
Sure. So the first paper that I took a look at was called Precise Genome Editing Underlines the Distinct Contributions of Mutations in ERG11, ERG3, MRR1, and TAC1 genes to antifungal resistance in Candida parapsilosis. So for a preface, Candida parapsilosis is a fungal yeast which is naturally found on the human skin. And under certain conditions or certain patient populations like ICU patients or preterm neonates, Candida parapsilosis can represent an opportunistic pathogen causing invasive candidiasis or candidemia, sort of deep-seated infections. And was identified as a priority pathogen by the WHO in 2022. So it's a priority pathogen because number one, it's capable of human to human transmission. We see it in hospitals. And two, there's a dramatic increase in the Candida parapsilosis that is resistant to the drug fluconazole. And why that's important has to do with the fact that fluconazole is a pretty powerful antifungal agent. It's used as a treatment for a variety of fungal infections. So unfortunately, the species parapsilosis, compared to other disease-causing species like Candida albicans or Candida glabrata, isn't really well understood in terms of the actual mechanisms behind the resistance that we're seeing to our antifungal arsenal. So it sort of segues into like what the paper is looking at. So from the title, I kind of quote contributions of mutations in the ERG11, ERG2, MRR, and TAC1 genes. And if you're not familiar with that sort of thing, what they mean here is that they've identified mutations or changes in the genes that are not necessarily random per se, but they're the mutations that might explain why fluconazole, the drug that we've kind of relied on for years, doesn't work well against these strains. So not just fluconazole, these changes, mutations could sort of like shed light in terms of whether resistance to one type of antifungal might be causing resistance to others. So instead of just observing the mutations, what makes this study really special is that the researchers used what's called CRISPR-Cas9 to precisely edit the fungus's genome. So CRISPR-Cas9 is a tool that cuts DNA at targeted locations and allows researchers to insert essentially synthetic DNA templates with specific mutations that they want to take a look at to create engineered strains for the study. So by introducing specific mutations, they could actually test how the changes that they implemented influenced the resistance to fluconazole, other azoles, and they even looked at amphotericin B as well. So you can think of it like flipping on and off a light switch and seeing sort of what works and what doesn't work. So the study really wanted to dig into three big questions. They wanted to know how much the mutations they looked at contributed to resistance. Were they specific to fluconazole? And then how does the genetic background of the fungus, things like the number of copies a gene has, affect its ability to resist treatment? And then three, is there a trade-off to these mutations? So just to talk a little bit about what they did.
Major findings now.
Yeah. In order to talk about what they did, they looked at ERG11, which is the actual enzyme that's the target of the drug fluconazole. And they found two mutations that really did reduce the susceptibility of fluconazole and caused resistance levels to increase pretty dramatically whenever there were two copies of the mutated gene, something we refer to as copy number. And second thing they looked at was the ERG3, which is an enzyme, or it encodes an enzyme that's pretty crucial for the cell membrane. They identified a few mutations there that made it more resistant to amphotericin B, and then also mutations that was called trailing growth, meaning that the fungus wasn't necessarily fully inhibited even at pretty high drug concentrations. The last two things they looked at were TAC1 and MRR, which are transcription factors that work on drug efflux pumps. And as we were talking about before, these are pretty important in terms of antifungal resistance as well. So they found a few mutations there as well that led to some pretty impressively high mechanisms of resistance. So I think the cool thing about this paper is that they kind of skip over the idea of using correlations to show whether mutations affect the resistance phenotype that we see. And they can actually use sort of a cause and effect model to show, like, in these isolates, we see the resistance associated with turning on and off, specific mutations.
Yeah, excellent choice, particularly because the technique pretty well advanced to just heal these mutations. So Lee, the fungus among us, right? Fungal resistance is a threat, particularly in immunocompromised patients. What are your comments on this?
Yeah, I mean, I think what's nice about this study is it's very elegant, and Nathan hits the nail on the head. Basically, they're not just looking at associations; they're using CRISPR technology to engineer these bugs to look at address very fundamental questions about specific changes in the genome, and that's what makes it such an elegant study.
Yeah, Bill.
Yeah, I echo exactly what Lee said. It's a very impressive treatment at the molecular level, and I would encourage my mycology colleagues in this area to publish molecular biology, molecular genetics manuscripts dealing with fungal resistance in AAC, because it's really the hot site for this work.
Yeah, and definitely we're learning much more about the molecular basis of this resistance, which is absolutely critical to understand and develop new drugs. Okay, from the fungal world, let's move to the parasite world. And then Taylor is going to tell us about the paper on parasite resistance.
Yes, so the first paper I'll be presenting today is titled In Vitro Efficacy of Next Generation Dihydrotriazenes and Biguanides Against Babesiosis and Malaria Parasites. And this paper is from a team of researchers from Yale, Princeton, and the University of Montpellier. And to be honest with you, I chose this paper because I recently was on anti-malarial medicine myself. So I thought, why not learn more about the subject? And unfortunately, I learned that these parasites exhibit pretty widespread resistance to current antiparasitic drugs. But that is where this research comes in. And the authors focus on developing next generation antifolate drugs that target the folate biosynthesis pathway, which is essential for the survival of these parasites. And the protozoan enzyme dihydrofolate reductase thymidylate synthase, or DHFR-TS, has long been a target for antiparasitic drug development, but efficacy of current drugs on the market is being challenged due to the development of point mutations in this enzyme. And those mutations impose spatial restrictions that decrease binding affinity of drugs. And so in this paper, they screen a library of 79 compounds against drug-sensitive and resistant Plasmodium falciparum and various Babesia species. And they did find a couple of compounds that worked out pretty well. So the compound called JPC 2067 and its prodrug, JPC 2056, exhibited inhibitory activity of Babesia parasite growth at the nanomolar level. And these compounds are actually analogs of the small molecule WR99210, which was developed in the 60s, and it exhibited really great biological activity, but it had poor bioavailability and caused gastrointestinal issues in healthy subjects. But now we have these analogs 50-something years later, which show little to no cytotoxic effect in human cell lines, which is certainly a step in the right direction. And they showed that the Babesia DHFR-TS enzyme activity is inhibited by the JPC 2067 drug, but not the prodrug. So they decided to take a look at that prodrug activity in vivo to see if metabolism of the compound would result in activation of the inhibitory function. And they saw exactly that. The prodrug delayed parasite emergence and reduced levels of parasitemia over the course of infection in mouse models. And it even completely cleared parasitic infection in a mouse model of severely combined immunodeficient model. So with really great results like this, they concluded by analyzing other analogs of JCP 2067 for anti-babesial and anti-malarial activity. And they did find one compound in particular, which they titled JCP 2060, which was the most broadly effective molecule.
Great. Yeah, I guess most people are familiar with malaria, maybe not many with Babesia, but it's another blood-borne parasite, mostly in the Northeast, the United States, that can produce severe infections, particularly in immunocompromised patients. Beautifully presented. You know, the 2024 is an alarming year because particularly malaria resistant to the artemisinin compounds have been described first in Southeast Asia and now in some parts of Africa, which is very concerning trend, particularly after COVID, because there seems to be a relationship between stopping of the funding from these programs and the emergence of malaria resistance, which really kills many people with no vaccine available yet, although there are several efforts. So finding new targets is a very important situation. So, Bill, any comments on this?
Yeah, I think this was great because the point you just made of finding new targets, how critical that is, but also too, it reminds us that things that might have been previously or anti-protozoal compounds that might have been previously put aside due to issues such as toxicity and bioavailability. Modern medicinal chemical approaches can bring these back to the forefront. So hopefully this will be an advance that will find its way into the clinic.
Yeah. Lee, so this is a purposing things that were before and then that found with the techniques that we have that we can really modify this. Any comments?
Yeah, I mean, this paper really hit home because I was on the clinical service, the inpatient service a few months ago and had my first homegrown case of babesiosis.
Oh, wow.
It was quite severe, and I was struck by the limited number of agents for treatment. And so anything that will bring new agents to market, I think will be very welcome.
Yeah. So great. So let's move now to the world of mycobacteria, another very difficult organism to treat. And it's Jacob McPherson who is going to present our first paper on this topic.
Hello, thank you. So the title of my paper I chose is Bactericidal and Sterilizing Activity of Novel Regimens Combining Bedaquiline or TBAJ587 with GSK 255-6286 and TBAJ7371 in a mouse model of tuberculosis. This was published in February of 2024. The first author is Si-Yang Lee, and the corresponding last author is Eric Nurnberger from the Center for Tuberculosis Research of the Division of Infectious Diseases at Johns Hopkins University School of Medicine. So there will be a lot of novel drugs, and this will be a little bit of alphabet soup, but the main reason I chose this paper is that multi-drug resistant and extensively drug-resistant TB is concerning when it's resistant even to the novel regimen that is preferred for it, and that is bedaquiline, protominid, and linezolid. And so this is a nice paper where they use a lot of mouse models of TB to study agents that will further build upon this regimen against those MDR and extensively drug-resistant TB isolates. And so they largely did their experiments. They largely did 4 experiments looking at what is correlated with these experimental compounds with bactericidal and sterilizing efficacy. And so I'm going to describe those 4 experiments quickly. So experiment 1, they wanted to look at the contribution of, I'm going to call it the compound G, as in Gary, and or compound A, as in Alan. So compound G and A in combinations to bedaquiline or protominid for bactericidal or sterilizing activity. And so they used the mouse model log10 colony forming units after 4 and 8 weeks of treatment. And largely they found in this first experiment, after 4 weeks of treatment, there was no significant difference between any of the 2 or 3 drug regimens containing bedaquiline or bedaquiline alone. However, after 8 weeks, the 3 drug regimen of bedaquiline, experimental drug G, and experimental drug A was more active than bedaquiline and bedaquiline A regimens, suggesting that the experimental A drug and perhaps experimental G drug contributes to the activity of the regimen. In experiment 2, they wanted to understand a little bit more about the PK/PD parameters with experimental drug A that was mediating this efficacy, this in vivo efficacy. And largely they used uninfected mice and found that after some series of dose-finding experiments, that the time above MIC was most correlated with experimental drug A activity when compared to area under the curve or maximum concentration, Cmax. Skipping to now experiment 3 of the paper, they wanted to evaluate the sterilizing activity of the bedaquiline, G, and A regimen with and without the combination, with or without the addition of linezolid to that combination. And they also compared this to other regimens such as rifampicin, isoniazid, and pyrazinamide as controls. And they found overall that after 4 weeks of treatment, the BGA regimen was significantly more active than the RHZ, or rifampicin isoniazid pyrazinamide regimen, but not the bedaquiline protominid linazolid standard first regimen for those resistant isolates. BGL was not as active as BGA at week 4, and this was statistically significant, and there was no significant difference at week 8 either. After 8 weeks of treatment, the regimen that was most focused on bedaquiline, experimental drug A, and experimental drug G, the regimen was still more active than the RHZ regimen, but was significantly less active than the first-line regimen BPAL.
Okay, and
so
the experiment 4 is?
Lastly, the experiment 4 wanted to evaluate the sterilizing activity of the G and A when combined with the next generation drug TBAJ 587 instead of bedaquiline. And so this is a novel, this is a next generation oxazolidinone now in phase 2 trials. And they largely found that after 4 weeks of treatment, the addition of G to this novel oxazolidinone significantly increased bactericidal activity, and when in combination when comparable to that seen with the oxazolidinone, protominid, and another regimen.
Okay.
And so largely they built
Sorry.
upon that.
Thank you, Jacob, for that detailed explanation. The pipeline for tuberculosis drug is increasing, and I would say it's exploding comparing to the was before. And this is very welcome news for the field. Due to time, I'm gonna move back now to Nathalie, and I'm gonna go back to gram-positive resistance.
All right, so the second paper I chose for gram-positive resistance is titled Molecular Physiological Characterization of the Dynamics of Persister Formation in Staphylococcus aureus. And this was work done by Shiqi Li and others at the University of Amsterdam. And so this article looks at the bacterial cells that remain after antibiotic exposure. And these persister cells, as they're called, have been implicated in recurrent and chronic bacterial infections. And so they're an important topic to study to kind of look at mechanisms of resistance that we could potentially target in the future. So the authors here used Staph aureus as a model. And so to look at persister cells, they exposed Staph aureus to either vancomycin, which is a glycopeptide, or enrofloxacin, which is a fluoroquinolone. And these two antibiotics have different mechanisms of action. And so this would allow the authors to see if the resulting persister cells may be different. So the authors looked at cells, bacterial cells, 1, 4, and 24 hours after being exposed to other antibiotics. And then they used proteomics and metabolomics to be able to get a look at the changes that the cells made to help them survive antibiotic exposure. And so overall, the authors found that there are many commonalities between the modifications that the cells made to survive pressure from the two antibiotics. First, they found that, first of all, that they were able to measure metabolic activity from the persister cells. They also found that both antibiotics led to persister cells that had more chaperones, increased cell wall biosynthesis, fewer ribosomes, and changed carbon metabolism. They also found some important differences in the persister cells from the two different antibiotics. So for the vancomycin-exposed persisters, they found that they had slightly increased chaperone expression compared to the enrofloxacin-exposed persisters, and that these vancomycin persisters were more active in their glucose consumption and acetate production. They also found that pathways that were enriched in these persisters were associated with biosynthesis of amino acids and secondary metabolites, carbon metabolism, and metabolism in diverse environments. They also found hints that, that likely the stringent response had been activated in these persister cells. And they found that most of these changes for vancomycin exposure occurred 1 hour after being exposed to the antibiotic. In contrast, the enrofloxacin-exposed persisters had increased expression of the ATP binding cassette transporters, or ABC transporters, which could potentially be involved in the efflux of antibiotics. These persisters also had enrichment of pathways involved in DNA repair primarily. Notably, unlike the vancomycin-exposed cells, most of these changes happened 4 hours after being exposed to antibiotics. And so overall, this study I thought was really interesting because of the wide range of data that they were able to collect. And although they weren't really able to directly test a lot of these hypotheses, they were able to generate a lot of hypotheses that future authors or future scientists can kind of take forward and test out down the line.
Beautiful. This bill, persisters, heteroresistance, tolerance, we're
Mhm.
and learning more and more about this phenomenon. We actually have a podcast with Dr. David Wise and Dr. Miller focuses on
Mhm.
heteroresistance, understanding this. But this is a hot topic, has become for a few years, and more in 2024, we are understanding more the molecular basis. Any comments?
Yeah, I really like this because it also points out that different antibiotics impacting persister cell generation. It gives us the possibility of maybe targeting different pathways depending on the antibiotic that could be used in the future in eradicating these persisters.
Absolutely. I think tools for this status will be very useful in the future. So let's move on to Alex for his second gram-negative resistant paper.
Okay, so my second paper is actually a really good a move from the first paper I discussed, and is titled Inhibition of Pseudomonas aeruginosa Quorum Sensing by Chemical Induction of the MEX-EF OPRN efflux pump. So from the first paper, we already talked about how Pseudomonas can become is can be less affected by even novel agents that are up and coming for antibiotics. So a completely different approach is also being considered to combat gram-negative bacteria like Pseudomonas, and that's called antivirulence. So where instead of we killing the bug with antibiotics which we know bacteria can become resistant to, what if we just limit the bacteria's ability to cause disease and to cause infection? And we can do that by using these chemical inhibitors that basically either block the function of or block the production of these what we call virulence factors, which are proteins and small molecules that bacteria uses to damage our cells and to cause disease and infection. So this paper is from the Biofilm Center from University of Copenhagen, which really does a lot of work studying how Pseudomonas is virulent. And so in this case, they're focusing on quorum sensing, which is a bacterial communication system that Pseudomonas has. So essentially, the way it works is when there's a lot of Pseudomonas in our body, they communicate with each other and they collectively turn on a lot of these virulence factors. And the goal of this paper was to basically shut that down using a novel chemical inhibitor. So they screened through a library of about 3,000 compounds and looked for chemicals that would downregulate the production of certain virulence factors. Specifically, in this case, they looked at the quorum sensing regulated ones, the elastase LAS B and the rhamnolipid synthase role A. Elastases are used to break apart our proteins and cause tissue damage, and rhamnolipids are used to cause cellular damage. So, and they came up with a lead compound. It was called ethylthio, and I have a hard time pronouncing this one, ethylthioenenon. And this compound was able to significantly downregulate these virulence pathways, and at the same time did not have any effect on the bacteria's ability to grow. And they were actually able to pinpoint a molecular mechanism to how it does this. And basically, it was able to do this by inducing this MEX-EF OPRN efflux pump that depleted the bacteria of its intracellular communication molecule that's involved in quorum sensing. So basically shutting the whole pathway down. So, and this is really promising because these antivirulence compounds that don't affect bacterial growth or kill the bacteria will have less selective pressure for resistance. And these are the molecules that may have a longer shelf life overall compared to conventional antibiotics.
Thank you very much. Yes. Lee, this antivirulence concept that that is evolving, hard to prove eventually in clinical trials because you have to be superior to standard antibiotics. But I think this is a good straightforward into antivirulence, very targeted compounds. Any comments?
Yeah, I mean, it's a very, very intriguing approach. I'm skeptical that we're going to eventually get to the bedside and have something that works, but I think it's something that's, given the problems we have with these really hard to treat, MDR bugs, I think it's worth a try.
Yeah, I am more optimistic than you are, but I understand the limitations. Okay, Nathan, let's go back to the fungal world and tell us about your second paper.
Yeah, sure. The second paper is a really great paper with a really exciting title coming out of CHUV in Switzerland. It's titled Exploration of Novel Mechanisms of Azole Resistance in Candida auris. And so, to preface, as you well may know, Candida auris is a pretty major hospital-acquired pathogen, and it's notorious for its ability to resist antifungal drugs like fluconazole and voriconazole. And resistance in the Candida auris is often linked in the research that we have to key genes like ERG11, which I mentioned before is the gene that encodes the target of fluconazole, or transcription factors that regulate efflux pumps, which I talked a little bit about before as well. So transcription factors that we know the most about are TAC1A, TAC1B, and MRR1. But this study actually set out to identify new mechanisms of resistance that are sort of outside of these already pretty well characterized pathways. So to do this, the researchers created what's called a knockout strain of Candida auris, where those 3 transcription factors I talked about were deleted. And it effectively removed the fungus's ability to use known efflux pump mechanisms for resistance of fluconazole. So they exposed their new knockout strain to increasing concentrations of fluconazole for 15 days, and it kept growing out multiple new cultures and allowing it to kind of evolve resistance in a natural way through alternative pathways than the one that we study. So that was really interesting. And they use whole genome sequencing, which is a technique that looks at all of the fungus's genes to look at sort of the genomes of their newly created resistant strain. What they found were a couple really interesting non-synonymous mutations, which means that just one amino acid was changed, but these mutations showed promise of being involved in some novel mechanisms of antifungal resistance outside of the ones that we study. So, for example, they found 2 mutations in a transcription factor called UPC2, which was found to upregulate the ERG11 and MDR1. Those are the target of fluconazole and the efflux pump I talked about before. And it increased resistance to fluconazole somewhere between 2 and 8 times compared to their wild type reference strain. They also identified a couple mutations in the UBR and MUB1 genes, which are involved in a pretty complicated pathway called a protein degradation pathway. And they end up sort of stabilizing a regulator of efflux, and it leads to fluconazole MICs, again, that were between 2 and this time 16 times higher than their reference strain through increased drug export. And the last thing that they looked at, which is really exciting, is a novel mutation in a gene called MRS7, which is a mitochondrial protein. And it sort of points to this potential for mitochondrial involvement in resistance, which isn't really very well understood and kind of remains understudied. So even though this MRS7 gene has kind of been thought to be associated with the expression of those efflux pumps I talked about earlier, the increased resistance that they saw by mutation of that gene actually persisted in knockouts of both MDR1 and the transcription factor MRR1. So it was potentially pointing to this pretty new, exciting mechanism that looks novel in terms of antifungal resistance involving the mitochondria. So to confirm those findings, they reintroduced those mutations into their wild type, the ones that they found to be significant, using CRISPR again. So this is sort of another application there. But the purpose was to sort of see whether or not the contributions held true. And for the most part, they did. And they found fluconazole resistance going from 8 to 128 fold higher MIC values than the original strain. So what I think makes this study really important is its attempt to sort of uncover entirely new ways that Candida auris is resisting treatment, particularly through like mitochondrial involvement or the protein degradation pathway that I mentioned. We really haven't done a lot to associate those things with antifungal resistance up to this point. So might open the door to some new strategies. And I think understanding what we don't understand is sort of the first step in developing new strategies.
Fascinating, great explanation. Bill, Candida auris, a very urgent threat, mechanism of resistance, like, some of the main bacterial superbugs, many different pathways for resistance. What are your comments on this?
Yeah, I really like this. And it really highlights, the very sophisticated molecular approaches that were taken here. And also to bypassing the contribution of drug efflux so that you can discover new mechanisms. I think that was critical. This was beautifully done, and I think it will be a major advance for our field, for the field.
Excellent. Taylor, tell us more parasites now.
Yeah. So the second paper I'll present today is titled Inhibition of AMPK Activation in Echinococcus granulosus sensu stricto Limits the Parasite's Glucose Metabolism and Survival. And I found this paper interesting because this parasite can cause infection not only in humans, but it's a huge problem for the livestock industry. And as someone from a small farm town, I want to emphasize that infections like these have the ability to not only impact individuals, but entire communities. And currently, the only way to fully cure disease from this parasite is through surgery. But of course, that's not always possible. And even if it is, that can, of course, lead to an increased risk of secondary infections. Anti-infective drugs are the most widely used treatment method as of now, but the drugs currently on the market and recommended by the World Health Organization have to be taken for pretty long periods, and they're prone to the development of resistance and side effects. So that's kind of where this paper comes in, with a focus on the therapeutic potential of targeting an essential energy regulation enzyme in E. granulosus sensu stricto. So the adenosine monophosphate activated protein kinase, or AMPK, is a serine threonine protein kinase, and it plays a key role in the regulation of energy metabolism. E. granulosus has to be able to get sugars from its host for its energy reserve. So the regulation of energy metabolism is pretty crucial for parasitic development and infection. And so in order to assess the potential for targeting that enzyme in this parasite, the authors investigated the effects of AMPK inhibition on metabolism. And something interesting to note is that there actually are drugs on the market that target AMPK, but they try to upregulate AMPK. However, in this paper, they are testing out downregulating this enzyme. And so they found that the knockdown of this enzyme in E. granulosus resulted in much lower viability compared to untreated cells. And this indicated that targeting the downregulation of AMPK could be a good treatment option. And so they began testing viability when treated with a specific compound, and it's just called compound C, which is a known inhibitor of AMPK, and it has been widely used to study AMPK signaling. So with 50 to 100 micromolar concentration of compound C, cells were completely dead by day 6 of treatment, compared to 53% viability with albendazole sulfoxide treatment, which is a derivative of one of the only 2 currently recommended drugs for treatment. And I also found it really interesting, their inclusion of SEM and TEM analysis of the parasites, which revealed very severe defects in the parasite's rostellar region, suckers, and body.
Okay. And TEM is transmission electron microscopy for people who are not very used to the use abbreviations. So echinococcus, a very interesting disease, sort of neglected disease, that usually present with cysts, in different parts of the body, at least in humans, and it seems to be in animals. And zoonotic disease. It's nice to see that people are doing research, very refreshing, and finding new drugs for this that are important problems in the developing countries. And as you, Taylor, mentioned, affect large communities. Lee, any comments on this disease?
Yeah, Cesar, you hit the nail on the head. I mean, it's a big problem in certain LMICs, and so having additional agents is definitely a welcome phenomenon.
Yeah, and we are seeing more and more in AAC these very elegant molecular dissections of these parasites to try targets similar to any other. And these are systems that are very complicated and difficult to achieve. Last but not least, let's go back to finalize these beautiful discussions to the mycobacterial disease. Dr. Jacob McPherson, please, let's finish this.
Thank you. Yes. So my first paper was on Mycobacterium tuberculosis, and my second paper is much shorter but no less important on Mycobacterium abscessus. The reason I chose this paper is because it was a nice investigation of serial isolates from an outbreak, where they saw several mechanisms of resistance that led to phenotypic observations. The title of the paper is Evolution Toward Extremely High Imipenem Resistance in Mycobacterium abscessus Outbreak Strains. And this is from research investigators at the NIH. And so a quick background is that imipenem beta-lactamases in Mycobacterium abscessus are a problem when using imipenem for the treatment of that MAC infection. And so this was an outbreak investigation of those isolates, and they found three main mechanisms of resistance that evolved stepwise, and they showed the MIC changes. And so the first change they observed in those isolates that evolved from the index case was a deletion of a porin MSPA that led to the first MIC increase from 8 to 32 micrograms per ml. And that was followed with a single nucleotide polymorphism in the ATP-dependent helicase HRPA that co-occurred with the second increase in the MIC from 32 to 256 micrograms per ml. And so they also observed a increase in the mRNA and protein activity, enzyme activity of the beta-lactamase BLA-MAB. And this was co-occurring with a mutation in a upregulator gene of the operon called PPIA. And they posit that this mutation in the operon regulator also led to the increase in the beta-lactamase activity in addition to its mRNA activity. And the investigators concluded that this work supports that carbapenem BLI combinations for treating M. abscessus that is imipenem resistant is important.
So Mycobacterium abscessus, one of the most difficult bugs that I ever treated, and in a very difficult patient. Bill, I mean, atypical mycobacterium are really a major problem. Combination therapy, intrinsic resistance, and it's very refreshing to see that, again, new targets are being identified, and hopefully we can have different strategies to treat these organisms. Any comments on this?
Yeah, I really like this paper, not just because of the real clinical significance of Mycobacterium abscessus, but also, too, the real importance of looking at fresh clinical isolates sequentially can really help define new mechanisms of resistance that might not be available if one limits themselves to just laboratory maintained strains. So I thought that was a very nice approach.
Yeah, and also the fact that we now understand a little bit better these beta-lactam resistance in these organisms, and how can we deploy potentially. We talk about these old beta-lactams inhibitors that were initially designed for gram-negative bacteria, but we can potentially be applied to these mycobacterial infections, with something that we're learning more and more, and will be very helpful.
Exactly.
Well, this has been wonderful. We have talent for the future. You guys have shown how very excited and very high quality papers that have been published in last year. I want to thank you for all your efforts and to be so succinct and give us the gist of the papers that you investigated. And Lee and Bill, thank you very much for your thoughtful annotations and editorial comments. So these are the 2024 best AAC papers of the year. Thank you very much for everything. This is Cesar Arias, editor-in-chief of AAC, signing off.